Hi everyone, how are you all? I am sure you are all good, and today I am discussing a very important topic, that is DNA fingerprinting. And about this, you all are aware of the importance of DNA fingerprinting in forensic science and also in the matters of parental relationships. So the great scientist who brought this concept was Alec Jeffreys, and later on it was introduced in India also. So, what is the concept and how it works? You have to all understand. So, first of all, a DNA fingerprinting means in the DNA, the human genome, the DNA has to be compared. But what to compare, why to compare? So, compare because you have to match the DNA sequence, the sequence of a particular part with the number of repeats to the other concerned samples. So, how do we do this? That is the matter of study or matter of concern. So, that DNA from Human Genome Project, we have known said that when you talk about DNA, 99.9% is same in all. Then how will you compare when it is same? So that 0.1% left is only the part of the DNA which varies. So in this 0.1%, the sequences are there of the nucleotides, and the repeats, number of repeats are there. So these repeats are actually the defining features or for comparison. So in this small part, 0.1%, which is called the satellite DNA, there is one part, that is mini satellite DNA, which is having the variable number of tandem repeats, VNTRs, of the sequences, and VNTRs are the sequences which are having different numbers of repetitions. So these repetitions are varying from chromosome to chromosome in the same individual, and also from individual to individual. So by these repeat numbers, you can compare with different samples. So how this is done? First of all, you have to separate the bulk of DNA with this satellite DNA. That can be done by centrifugation. Okay, when you get the 0.1% satellite DNA, then you have to restrict the areas by a biological seizures, or that can be done by the process of restriction nucleophase. So that is, restricting the specific area. Now once it is done, you get the fragments, single stranded fragments of DNA. So these fragments again have to be separated because they are all jumbled up now. So that is done by gel electrosporosis. So electricity is passed through this, and a plate is there with anode and cathode. So when electricity is passing, so what is done? What is happening? The smaller fragments, single stranded fragments, will go upwards in the upper area, and the bigger will settle down. So according to the size, they can be separated. These fragments can be separated. But the gel is so weak, the base is so weak that it can destroy very soon all these fragments. So what can be done? It can be transferred to another semi-membrane, synthetic membrane, that can be nylon also, for their security, for their protection. And then after you have transferred this, so but we have not seen that what is the position of these fragments. Now you have to then hybridize these fragments with probes, which artificial fragments of these variable numbers of tandem repeats, VNTRs. With these VNTRs are also there in the original plate, and also we are attaching them with our probe, that is artificial VNTRs. Now when we have attached them, These are radioactive VNTRs, so they can emit some lights also. Because up till now we have not seen, the scientists have not seen where these, after attachment, what is the position of these fragments. But after these radioactive probes, the artificial copies, when they are attached, then they are emitting lights. So they are showing the position of different fragments, up to which distance one fragment has moved, and up to which distance the another fragment has moved. So like this you get a pattern. When you get a pattern, you can compare this pattern with the suspects. This is the sample of the crime site. Now you can match with the suspects. The pattern of the suspect which is matching with this, the sample of the crime site, then he is the criminal naturally. So like this the decision is taken, the conclusion is reached. So by this process you can this fingerprinting is done, DNA fingerprinting, and you can just understand the process very well when you just go by step, and how this is isolated and then restricted, and then your gel electrosporosis is done to separate the fragments, and then these fragments are then again hybridized. So ultimately what we get, we hybridize them. Well, the original your frequent number 10 of repeats VNTRs with the probe VNTRs, and then we see that by the lights that they are emitting, they are radioactive probes, so they are showing the position of the fragments in the plate. Okay, and this position of the fragments, smaller and bigger, up to which distance one has moved, the fragments have moved. So like this pattern we get, and we match them with the suspects, and we reach to a final conclusion. So this is how it is done. So I have just talked in a gist. I think it will help you in understanding the process. Thank you very much. God help you all. God bless you all.